Next up is someone who probably needs no introduction, but I'll still do it anyway. She holds a very special place in our community story. Dr. Erin was one of the first scientists to connect the dots between mutations in the SON gene and what we now know as ZTTK syndrome. Her lab at the University of Alabama, Birmingham, has been studying SON for over a decade. And her passion for this work is deeply personal. She's not just a researcher, she's a founding member of this foundation, a member of our board, and one of the key reasons we know as much as we do today. Please join me in welcoming Dr. Erin on.
Hello, everyone. I'm so honored and happy to be here. So yesterday, I first met many of our board members and parents, researchers, and I was really overwhelmed and so excited, and I couldn't fall asleep last night. So I ended up having just a 30-minute sleep in the morning. But I'm still energized today by looking at all your faces and your all sunshine. So I'm extremely happy to share this moment with you. So we've been waiting for this day for many years, and then finally the day is here. So today, my talk will be for you, parents, family, to understand more about ZTTK syndrome and underlying biology. So let's get started. So we'll talk about ZTTK syndrome definition, clinical phenotype. Then we'll learn more about the gene SON and the underlying pathology on ZTTK syndrome. And then I'll introduce some research model we have, mouse model, cell model, and then we'll introduce our current future research, and then I'll also take this opportunity to introduce the people behind it. So first, the ZTTK syndrome. So as you know, ZTTK syndrome is an ultra-rare neurodevelopmental syndrome. So that means this syndrome affects brain development and also actually many parts of our body. So let's just break down. First, ultra-rare means we have really a small number of patient cases so far. So only around 80 patients has been officially reported in the literature so far after it's first published in 2016. And we have over 300 patient cases, thanks to the foundation and then Facebook group. But we hope we can find more patients all over the world. And this syndrome is monogenic. So that means this syndrome is caused by a change in a single gene. So in this case, the SON gene. So the SON gene is on chromosome 21. So chromosome 21 is also involved in Down syndrome. So we'll look at more about the biology later. And the ZTTK syndrome is neurodevelopmental disease. So that means it affects the development of the brain and the nervous system. But actually, we know it affects other systems too. And the symptom, in many cases, to develop in infancy. But we also know some other cases that we saw some regression. That means the child can, for example, speak previously and then lose ability later. So we are still working on this, and then your participation of all these symptom surveys are extremely important. So far, we don't have known cure or treatment, but we are here to change it. So this is a snapshot of how our knowledge has grown over the year for ZTTK syndrome. So it all began in 1988. One Russian researcher group reported some small fragment from the SON gene. So but that time, they didn't know what the SON gene function in the cell. So after that, almost like 23 years, it's a very few research on SON gene. And until 2011. So in 2011, so we published that the very first time SON function in the human cell, so which is my postdoctoral training research. And then later, 2015, when exome sequencing, genome sequencing are available, the one group showed that they found the SON gene change in the rare disease patient. Then after that, 3 groups, including my research group, showed that SON gene change is really linked to the very unique set of clinical symptoms. And then these symptoms later named as ZTTK syndrome. So in 2021, Dingemans and his colleagues summarized clinical feature from 52 patients. So this report is so far the most comprehensive summary about ZTTK clinical symptom. So this paper described several information. And then first, the ZTTK syndrome show any truncation variant in the SON gene. So this is a very important concept. Actually, I'm going to explain about this later. So we'll skip at this point. And then many cases, we found this mutation in the SON genes are de novo. So that means these changes are new in the child, so which are not inherited from the parent. And then many of those genetic change has been only seen like one single cases. So that means each patient has a very unique change in the SON gene, but one change over there showed the specific with number on around 25% identified ZTTK patient share the same genetic change. And this affect both male and then female, and then so far we know most of them are children. So most prevalent phenotype, the commonly identified phenotype from ZTTK individuals are intellectual disability and then neurological abnormality. So intellectual disability we saw from severe, mild to moderate, those different degree of severity. And for neurological abnormality, around 92 to 95% to show patient show this neurological symptom that include hypotonia and then enlarged ventricle. That means the enlarged fluid space inside the brain, and then seizure. And then some of them show the very disorganized cortical layer, which is the outer layer of the brain. So those can be diagnosed by MRI screening. And then beside this intellectual disability and neurological symptoms, we know the ZTTK individual experience many different type of symptoms. So first, over 90% of individuals show facial morphology difference, and then neurological, of course, and then neuromusculoskeletal, so muscle and then bone issue, and then GI issue, and then behavior problem. And also we know many ZTTK patient showed the visual impairment, heart, and the immune system change, and then metabolic issue. So all of these could be changed, can be changed in the future if you participate more clinical survey. So if you think about ZTTK syndrome like this ocean, we just look at the surface of this ocean. But to understand this disease, we have to dive to deep inside the ocean. So that's what we are going to do. So let's dive into the biology with me. So in the previous session, Raymond introduced this concept, gene and protein. So gene is the recipe to make a dish, and then protein is a final outcome, your dish. And then actually there is one more step between this, it's called RNA. So think about this case. If you have a cookbook recipe, so this is your book, but you want to make your own memo, so you just make your note and then get just a copy first of this cookbook, and then, okay, you understand, and then based on this, you make dish. So that's a final product, protein. So if you think about the son case, first we have son gene on chromosome 21, because we have a pair of chromosome 21. We have 2 copies of chromosome 21 in most, not all actually, of our cell in our body. Then the son gene makes son RNA. So you have to first make your own memo. So this RNA then can be good reference to make a protein. So in our cell, almost all part of our body cell type shows some RNA and then some protein presence. But it's very interesting. If you see this RNA level and protein level, you can see RNA and protein level do not always correlate. So some cell types, some organs show the same amount of RNA but different amount of protein. So that means there is also regulation step between RNA to protein. But then you can see the son is expressed in the many, actually all cell type in our body. Then we human had son in all type of our body cell. Then how about other organism, other species? So son is very conserved. That means other species also had son or son-like gene and protein. So first, we have very long, large son protein in our cell. And then mice also have similar type of son. But if you see these other species, zebrafish, fruit fly, and then worm, they have a very simple version of son. But surprisingly, look at this froggy son. It's huge. So froggy win. So this information give researcher the idea of which part of the son protein is functionally very important, which part could be more flexible, and then also this can tell us which animal model will be best for the human ZTTK research. So now we will learn about the function of son protein. So when our cell make son protein, the son protein should be somewhere inside of our cell. So when we look at the cell, we have the compartment called the nucleus. So these are the place where we have a chromosome, genomic DNA. Then this genomic DNA actually interact with a lot of protein. So when we do specific technique to visualize where some protein is inside the cell, you can see this red spot. So it's just an irregular patch. It's like a freckle in the nucleus. So we call this nuclear speckle. So this is the main location of the SON protein inside of our cell. So you can see this red nuclear speckle where a lot of SONs just gather together over there. And then you can see the green part actually represent our genomic DNA. So they are very close to the genomic DNA. And then what this SON protein doing over there? So let's go back to DNA, RNA, protein story. So we learned about RNA. We need to make a script based on recipe. You have to first, make your own note. But when you make this note, first you just copy everything from the recipe. So that actually includes something that are not very important, such as take 3 steps forward, open the refrigerator, and then grab 1 egg, or go to the pantry. That take like a 5-step thing. So those are not that important for your final recipe. So those parts that are not very important are called intron. Then shown here as a linear line. And then very important information for your cooking is called exon here. So when you first make this note, you have both exon and intron part. But to make a good outcome, you should have really simple, important information. Otherwise, the other part just distract you. And then you sometimes just mess up because of those distractions. So those intron removal step is called RNA splicing. So I didn't explain the very first part. You have to make this recipe note called RNA. So making this RNA based on the DNA, the recipe information, this process is called transcription. So this transcription and RNA splicing are 2 main functions of the SON protein inside of the cell. So when you see the nuclear speckle, the SON is there, and then they are very close to DNA, and then doing its job for transcription control. So in the control means sometimes it can really make it better, make it more, or sometimes it block and then make it less. And then also when the cells make this RNA with intron, the SON should be there to remove this intron properly to make a final product, to make a good protein. So these are 2 main functions. And then when you think about the ZTTK syndrome, so we have 2 copies of SON gene. And from 1 copy, we can find a DNA sequence change. So that happened only in 1 copy. So we called this heterozygous mutation. That means it just happened in the 1 out of 2 copy. So we have still 1 normal copy of the SON gene. So it can produce SON protein, but you have only 1 source. So the cell of our ZTTK patient, our sunshine, has only 50% of the normal level of SON protein. So that affects SON function in transcription and RNA splicing. And then we know lots about its effect on certain functions in RNA splicing. Because when you have enough amount of sun, it can remove the intron properly, efficiently, faster. But if you have only half of the sun, this intron removal step is not very efficient. So you have sometimes those intron retaining. So your node that containing not very necessary information. So then you can distract you later. So then how these sun function change link to the clinical symptom we found from ZTTK individual, our sunshine. So I will give you just the 3 examples. There are a lot of underlying molecular mechanism, but we'll make it simple. The first, the sun target DNA, RNA are very important in our brain development. So if you can see this like a middle part diagram, during our brain development, our neuron should migrate from inner part of the brain to the outer side of the brain. That's called neuronal migration during development. That actually put all the neuron in the proper site in the brain and make a good brain structure. But in this process, of course, there are many proteins involved, and then some of those include TUBG1, tubulin gamma, and then mRNA, filamin A. Those proteins are very important to support the neuron to migrate to the proper site. So without the good amount of TUBG1 and FLNA protein, then this neuron cannot migrate properly. So when we have only half amount of sun, those RNA processing for these genes actually is affected. So you can see this, the experimental data, it's called from the PCR. So this PCR experiment can detect whether the cell has intron removed RNA or intron containing RNA. So if you see this upper band that contain intron, so this is long, so actually in this PCR and then gel, the image you can see is this upper band. And then you see this lower band, that means no, this product doesn't have intron, so intron is properly removed. So when we isolate the blood cell from mother, father, and then 2 sunshines, and then check out this intron containing and remove the product for these 2 genes, then you can see this ZTTK cell contain more intron containing product. So that means the less TUBG1 mRNA protein, then they cannot support the neuronal migration properly. And then also I will give you the couple of examples of those genes which are very important for our metabolism. So we have the metabolism in carbohydrate, lipid, protein. Then first, those nutrients we use to generate our cell's energy, called ATP. So in this process, of course, the pathway, the biochemical, very complex biochemical pathway involved. So one of very important part of our body to generate this energy is the organelle called mitochondria. So inside of a cell, not in the nucleus, the other part is called the cytoplasm. Over there we have mitochondria. That's like the powerhouse of our cell. And then they have special the reaction called the TCA cycle, electron transport chain. That is a very important process to generate energy, ATP, for our cell. So in this process, you can see the called IDH2. This protein is actually enzyme. It's facilitate this TCA cycle. So without IDH2, you can imagine the less TCA cycle and then less electron transport chain, the supply from TCA cycle and then less ATP. So that's what exactly we can see from ZTTK syndromes, the patient cell. So we see the IDH2, you can see the same PCR data show IDH2 RNA contains more intron when you look at this from ZTTK patient. And then the other enzyme we know as a zone target is ACY1. So this one is important for protein metabolism. So protein is actually made out of amino acid. So the amino acid are the building block to build the protein. So once you have amino acid and then make this protein by putting them together, for many cells function, this protein should be decorated by other modification we called. So one of these decorations is acetylation. I just mark with AC. So this decoration later should be removed when we want to recycle our amino acid. So if we don't have much the nutrition resources, our cell can break down some protein and then reuse this amino acid. So this process, this decoration AC should be removed. So ACY is playing a critical role in removing this AC. So if we don't have enough ACY1, then you can imagine these decorations are not removed properly, and then amino acid recycling is not that efficient. So this ACY1 is also zone target, and then you can see intron-containing products are increased in ZTTK syndrome cell. So these are the some of the example we know, and then of course, we have actually in our cell around 20,000 different genes for protein coding. Actually, not all of them are zone target, but the really good number of the gene need the protein zone for their proper transcription and then RNA splicing. Then we're going back to these questions I skipped in my previous slide about frame shift nonsense. Probably you heard about this terminology from your genetic report. And so those are called the truncating variant. But besides this, we can see also missense in frame deletion. So what are these? So what kind of change we can see in the zone gene? So to explain this, let's learn about reading frame. This is a very important concept to understand the ZTTK pathology. So in our cell, when we make a protein, our cell read RNA letter and then interpret this into amino acid, which are the building block of the protein. So during this reading process, our cell read every 3 letters in the RNA. So this is called reading frame. So 3 letters make 1 amino acid. So let's get one simple example. Because DNA, RNA use this only 4 letter ATCG, in RNA case AUCG. But let's think about in our alphabet case. So when you combine 3 alphabet letters to make 1 meaningful word, so you can see this example, the big red dog. I'm a dog person. I really like a dog. So if someone asks me, oh, what do you want for your birthday? Okay, I would say the big red dog. Then you can get the dog. Okay? Then if you miss one letter, so here, if you miss H in the first word, then it's just so messed up. Right? That means nothing. So in our cell, there are more letter continues, and then at some point, you can find the combination of alphabet like E and D, and then our cell stop making protein, making amino acid. So in that case, we called it frame shift because 3 letters make one frame, but you really change this frame. So that really changed a lot of protein composition. So you cannot make proper protein. That's frame shift mutation. So let's learn about nonsense. So if you join E and D, now our cell recognize this. Okay, stop. So in the actual case, it is not the E and D, the other letter. So in this case, if you put the big and then E and D, stop. And then, no, you cannot get a dog. So, no, this is a problem. So that is called nonsense mutation. And then we have in-frame deletion. So the frame is 3 letters make one frame, right? So it's just in-frame means just a 3-letter deletion. So you can just miss some of those words. So the big dog or the red dog. Okay, if you don't care about it's red or big, they'll be fine, right? But I really need a red dog. I don't like a blue dog or black dog. Then it is a problem. So in this mutation, those frame shift and nonsense are definitely loss of function mutation. But in frame deletion, sometimes it could cause loss of function, sometimes not. And then let's think about different case called missense mutation. So missense mutation means you still have this reading frame, but you just show a little bit different meaning. So let's think about this case. You change the last letter G to T. Now it becomes the big red dot. Oh, this red dot cannot be a good pet. Don't think so. So this is a loss of function. And then in some case, if this thing happens somewhere, and then this missense mutation, then it can be also loss of function mutation. But in other case, if you change D to H, it becomes the big red hog. I like hog too. So if you don't have a dog, I can keep this hog as a pet. So it's a missense mutation, but it's very different from the big red dot. Right? So that's why if we have missense mutation, it is very hard to interpret whether this causes loss of function, whether this is ZTTK syndrome or not. We should decide based on where it happened and then what's the outcome. So if it really make a dot, it's pathogenic. If it make hog, and then this hog can be doing same thing, it can be a cute pet, and then no problem, then it is not loss of function mutation. So it probably little bit affect your preference. It's a hog, so no, maybe it's not the same as a dog, but it can do some part of its function. So if you look at your genetic report, you saw a lot of the genetic medical terminology. So if you see the letter C, that means coding DNA. So DNA sequence for protein. If you see the letter P, that means protein. And then the letter FS means frame shift. So we learned about the frame shift. And then this star shape, that means stop. So these are very important part that you should know to understand your genetic report. And then this diagram shows the location of the SON gene variant. We have identified over around 70 patients here. And then you can see they are scattered all around the SON gene and then the corresponding SON protein. And then we saw this one spot that show around 25% of identified cases. So in this case, you can see C, and then this is the number. So that means this is the number in the context of coding DNA. So if this DNA is the recipe for protein, right? So if you want to really see what happened in the final product protein, then you can see this P and then some letter. So those alphabet V, E, or sometimes it shows that BAL, GLU, those represent amino acid, the affected amino acid. And then those number mean the location. And then FS means what? Frame shift. And then star means the frame shift code the premature stop. So then those, the letter after stop means abnormal amino acid generated after due to the frame shift. Okay, so one of the questions we have heard from many patients are these different mutations has correlation with clinical symptom and then outcome? So far we think if those are frame shift, the nonsense mutation, which are definitely loss of function, in this case, we think the location and the phenotype really do not show any correlation. So I'll explain to you why. So our cell has a very smart mechanism, the quality control mechanism. So if cell produce the RNA that show the stop message in the wrong location, we have a quality control mechanism to get rid of this RNA. So if you see this red line, it's just SON RNA, and then you should have a stop message at the end of this RNA. Then it can make entire full-length protein, SON protein. But in frame shift and nonsense, we have a stop in the wrong place. Then our cell recognizes this. Nonsense. Let's get rid of this RNA before it make something wrong. No. So the process called nonsense-mediated mRNA decay, NMD. So due to this process, those RNAs are all eliminated, and then they are not participating in protein production. So there are some exceptions. If those wrong stop location is very close to the actual stop location, maybe around the 50 to 55 RNA letter distance, then our cell can say, okay, you pass. Go and make protein, and then make slightly truncated protein. But so far in our ZTTK syndrome patient, the location is not very close to the actual stop codon, so we think most of them goes to this nonsense-mediated decay process. So next, I'll briefly introduce our ZTTK research model and then resources. So first, cell model. So most important and then the best model will be the patient-derived cell model called here iPSC. So this model, we should get the cell from sunshine, the patient, and then those cells could be blood cell or the skin cell, and then we can reprogram this cell to make this stem cell. And we called this induced pluripotent stem cell. It's called iPSC. So this iPSC has great potential. So if we put some signal, then let them go to different type of cell, then we can make neuron, adipocyte, blood cell, and even sometimes we can make small organ mimic. So these are really good resources for the ZTTK research. And then we're really happy to announce that we just made the world first ZTTK patient-derived iPSC, and then they are really ready for our research. And then mouse model is also a very important tool because mouse son and then human son are very similar. So we generated the mouse model. So this model have really 50% of son, just to mimic our human ZTTK syndrome. And then you can see they are smaller than other mice. And then we saw they have very low fat content in the body. And then when you look at their muscle fibers are very tiny. So these are really great model to study many parts of this body organ. And then when you put them in this open area, and then when we look at where they go, they move around, and then track them, then you can see the GTTK mice really stick to the edge of this open space. They are not going to the center, so that actually represents some anxiety-related disorder. And then when you look at the bone, you can see the lower bone density. So we can use this model to study multiple organs. Then we hope to find many other mechanisms in different organs. So our current and then future research, we are using this mouse model to study many different organs. And then currently we are really focusing on the metabolism because those mice show very different fat cell, adipocyte, and then liver function to process their lipid. And then we are also interested in immune system study. What kind of cell the change in the immune system? We think their B cell has some problem. So it's very important for the immune defense and then vaccine response. So it's exciting progress. And then we also study muscle, and then we plan to do more the neuron study. And then for the cell culture model, we still have a lot of questions to answer. So what are the direct son DNA and RNA target besides those few examples I explained today? And then son is really long protein. And then you can see some lower organism has very simple son. Then which part of the son is most important? Now that could give us really important information for therapy. And then beside the transcription, RNA, we think son has more function. So we are continuing to study those unknown functions. So we are also working to move forward our knowledge to the therapy development. So this is the big support from foundation and then our scientific advisory board. So how can we develop therapy? The first, remember we have one copy of normal gene, the other copy is affected. So then first, can we get two normal copies by just to fix this DNA change? So just to fix it, that's gene editing. if you cannot fix the right spot, can you add the new copy? That's gene therapy, the adenovirus-mediated gene therapy. And then also, we know what is most important thing here is not some gene, actually some protein. The protein is very important for our function. And then we know to make a protein, so the gene make should RNA and then protein. So this process can be regulated by something. So can we regulate this process to make more RNA and then more protein from one normal copy? So this is our effort to find the activator of this transcription process to make RNA and then protein production process. And also, as I mentioned, the most important thing here is some protein and some target gene, some target RNA, and then some target the proteins made by those some target RNA. So that's why we have multiple symptoms here. So for example, we saw the metabolism change, right? And then energy insufficiency. If we don't have enough energy, can we find better way to supply more energy? So those are just the low-hanging fruit. We can just identify the very important finer symptom. And then while we are working to fix the original root cause, we can do something in the short time, like to change the final outcome. So that could be, find some drug that can improve the symptom, and then also the nutrition change, lifestyle change. That can be really good management strategy. So I'm going to take probably next couple of minutes to introduce our lab. So we are at the University of Alabama at Birmingham. So this is the building where our lab is located, and then these are the picture of the space where we culture the cell, where we look at the DNA, RNA. And then those research need a lot of material, and then time, effort, equipment. So we also get really good 2 pieces of equipment from Thermo Fisher, and then supported by our ZTTK Sunshine Foundation. And then we are really excited to use this tool to progress our research. So these are our current members. So we have many PhD program students. And then Amar Mustafa work on a lot of GTTK model, and then to find activator of the zone protein and then RNA production. And then Kayla and Sophia work on more basic mechanism. And then Leah side, they are working on immune system and then the iPSC. And then Hyung is the lab manager. And then we are also very happy to recruit a new researcher to start our neuron study. So this postdoctoral researcher recruitment will be supported by our foundation. So I'm really grateful. And then we have also former members who are greatly contribute to our previous research. So ZTTK, K means the last name Kim. So the Jang-hyun Kim is the person who spearhead our first discovery of GTTK and then identification of a lot of some target RNA. And then Lana established the mouse model, and then Bohe did a lot of work for GTTK metabolism study. So all of our work are for you, family, and then our sunshine. So these are the reason why we are working. So thank you so much for sharing your story, for being here, and then being together here to have this time together. And then I can't explain how thankful I am to our foundation, make all of this happening. And then we have a lot of collaborators shown here. And then we will continue and then work hard to make progress until our sunshine shined even brighter in the future. Thank you so much.
Thank you so much, Dr. An. We'll skip Q&A, but she'll come back on stage later this afternoon. We love you, Dr. An, for your dedication, your leadership, and for helping us understand not just what's happening, but what is possible. Thank you so much. Another round of applause for Dr. An. Yeah.